Okay. Well, good morning, everyone. Welcome to the second day of our workshop. Today we'll begin with discussing about how we want to proceed with CAMI in the future. So you've seen yesterday in the talks that something that has been put in place is also a framework which can be used for further competitions. So from now on, things could become more and more routine, and we could provide further challenges. And there's also simulation pipeline software available, so we can also create new data sets. We are in discussion with several experimental labs who will be happy to also provide further unpublished sequence data sets. So there are the opportunities to make new challenges for the community. And we want to get your input also about how to best do it. We will have some talks about this now in the following, both via Skype, one from Aaron Darling from the University of Sydney, one from Ninja Nagarajan from Singapore, from the Genome Institute there, and one from Phil Blatt about computational support that the Pittsburgh Supercomputing Center will be able to offer to challenge participants. And then later on, we will also have a talk by Todd Triangen, who will be presenting challenges in a different application yet, namely pathogen detection. So this is something, at the moment, we focus on assembly binning and profiling, but of course there are other things that people really want to get out of the analysis of this data set, and we could move towards such challenges also in the future. A principal question, for instance, could also be, at the moment, this first challenge has been organized in such a way that we provide data set, participants submit their results and tools, and then we all sit together, evaluate them, and publish this. Individual developers that want to publish their tools, of course, have their own agendas also and their own timetables. So they would like to benchmark their tools on some informative data sets in an easy fashion, and then probably immediately proceed to publishing those results. So an alternative, what we could think about is if, in addition to such challenges where a larger group of people publishes together, we could think about simply providing interesting data sets, and then immediately after the challenge ends, people get their results and can use them for a paper along with the respective benchmark data sets. So this would have the advantage that people integrate their tools into the system. They can immediately use it for their papers, and the number of tools in the database will continue to grow, and it will allow us just to get, hopefully, a really nice overview easily over the state of the art. Yes, so we thought that maybe before we start with the talks of our invited speakers, we could collect some of your thoughts onto what you would like to see. So from the people here in the audience. And yes, so we would just take down some notes on...
And you're welcome to edit the document also. So if you don't have ideas right now, just think about it, and you can always edit the document and add whatever ideas you might have. So,
I know people have some thoughts, so just throw them at us.
Okay, so I mean, I'll talk about this later on, but under technologies, I think, so I'll talk about how we could perhaps provide a service for people that has computational support in addition to the tools that CAMI will provide, the Docker and BioBoxes and these sort of things. And so I'd like to explore how we can integrate that with large-scale computing resources and provide maybe some sort of a web service gateway to the computational resources where people can upload their data, and then the pipelines have already been installed and optimized on high-performance computing systems, and they can run the Docker containers and get results, and they can even then run the analysis, the evaluation pipelines directly on their results, and do it not only for sort of small data sets, but very large-scale data sets that they might want to explore.
I like it because we had people that were unable to submit because the time wasn't there, or the computational ability. They could simply submit the Docker and we calculate it for them.
So then we would send it off to PSC and they would calculate.
Right. So we could establish some sort of community resource there, so, which will require some work on our part. But we started this last time, and we'll be able to build on that.
Yeah, that'd be very nice. I mean, so the idea would you want people just to submit the tool and you would do the rest? So we would move away from people submitting results, or?
Well, they would be able to run their pipeline through the resources at PSC. Is the idea. So they would still be, the researcher would still be involved in running it, but we may be able to provide. We would, as a group, we would have to do some work to provide the interface. How would they do that? And so we already have some interface, through the CAMI. What's already been developed, so we could post that at PSC, and then have some interface for them to actually run the jobs there, either directly or through web interfaces like Galaxy or things like this. So we'll have to talk about how exactly we want to make that available, but there's some different opportunities.
Okay, yeah. So Phil Blatt is a person to speak to about these things. If people have requirements for high computing resources. Yes.
On the data sets, in addition to pathogen stuff, and that could be sort of discovery of pathogens, you're trying to tease apart, say, human from pathogen. There's also an issue when you're trying to look at the microbiome that you have a choice of going in, say, looking at 16S or doing some unbiased sequencing approaches. And it would be nice to be able to understand how the tools sort of match up between those things. So if you had a simulated data set where you had some bacterial species and then you could produce 16S from that, as well as sort of the whole genome and so on, that would be a good, that might also help.
So you want to quantify, or basically do a benchmarking. It's common in the field that people do both, right? And to see how...
Not necessarily, no, they just don't have the funds normally to do both.
Yeah, that's
so they make a decision, I'm just going to look at the microbiome and I'm just going to do 16S, or I want to capture, for example, bacterial phage relationships, which are becoming increasingly important in understanding the microbiome, and then they go to the whole genome sequencing instead.
You want to look at the 16S for pathogen discovery, if that's valid?
No, this is separate. So if you're looking at a bacterial microbiome, sort of how do the tools sort of match up if you're either doing 16S or whole genome sequencing, and what can you get if you try to merge those two?
Right, yes. We thought about a 16S challenge, but it's not so easy in some ways. I remember that it wasn't so easy to define it. Well, with 16S you usually do these OTU discoveries, right? These are, well, the whole species concept is kind of vague. And maybe one could do it, but I think we would have to put more thought into it, how to best design a 16S challenge. So the 16S, yeah. More thoughts on things? Yeah.
So besides users submitting new tools, one thing that should be kept in mind is as new metrics come up, being able to create some sort of framework so that somebody can define a new metric and then also sort of submit that to be able to run it against all other existing tools.
So the new metrics. Do we have like the metric, like the bio boxes, the analogous thing for metrics?
Yeah, we have the definition for an evaluation container.
Ah,
So
okay. So I know that there's been requests that you want more data sets that are like multi-data set, multi-sample data sets for those covariance-based spinners, for instance, right? So those tools, they would. So what would be your thoughts on that? Like how many data sets, how many samples should such a data set consist of? Should one make several, like 10, 20, 30 sample data sets?
Well, I heard more like 50 to 100.
It depends on the community complexity.
So for the high data set, what do you think? Like our high complexity data set, like how many samples would you need?
It depends on how you simulate the covariance.
Okay.
So if you do a new challenge, what would you want?
I think that's the question.
You don't really know.
The
So
CAMI can maybe can answer.
So
So you would like to answer the question like how many data sets do I need for this particular complexity to get the best result?
That's the reason it's not so 50 data sets is not realistic, but maybe that's what you need to resolve it. So
So one issue we thought about though, I mean, if we have one community, and then we provide like samples from that with 10 data sets, 20, 50, then of course people could also pool the data sets and then submit a result. But I guess if they submit their containers, we can reproduce it or run it. There was a question like for a contest, and some might use all of them for the results, some might just use subsets, and so that would make it hard to compare. But if submitting tools, that wouldn't be an issue. So we could explore that. So for a complex data set, maybe do like a whole, or a complex community, do a whole series of different data sets. Is that relevant? You look doubtful. I'm not sure.
Johannes, you said it's not realistic. You mean from computational?
no, I mean it's
From cost,
from
the
an
cost point.
experimental point of view. I mean the variance, like the abundances that we simulate, are more or less random, so they are very good for covariance things. But in practice, I believe that they don't differ so much. I don't know.
Maybe for a lot.
They differ a lot?
Yeah.
Okay. Then it becomes easier, but still, I mean, it's not realistic to have 100 samples of 15 gigabase pairs, I think.
No, but there are
pretty
centers that
expensive.
do it, right? Like big
Yeah,
sequencing centers
big
do it,
consortia.
and it would be interesting to see if this is the way to go for the future.
Depends on the question you want to answer.
Well, yeah, of course, it depends on the question. It depends on
Basically
like your budget.
wanting to get very specific, genome gains.
How about meta-transcriptomes?
Yeah.
Would tools do binning on meta-transcriptome? What kind of challenges do you think would be most relevant for meta-transcriptome analysis?
Something that's not specific to metatranscriptomics, but is visualization. So visualization of all this data. So there are some nice sort of JavaScript platforms that you can use and make a web-based dashboard, because trying to put everything on a slide at one time is just overwhelming in many cases, as we saw yesterday with the results of the profiling. And so something that's more sort of where you can explore dynamically would be for all the challenges, not just the...
Yeah, it was really nice what David showed, his idea also with the ranking and the scaling. So I don't know if you discussed already if it's possible to put that in the framework. Yeah, cool. That'd be nice. Would we go along what you're thinking, or with the with the?
Yeah, the sliders. Yeah. I mean, just having as long as one has sort of a uniform output for a particular tool, then it'd be straightforward to write a JavaScript thing which would just consume a JSON formatted file and then allow you just to have drop downs and so on.
Well, just as you said, when you look at all the data and all these different plots, it's really hard to put everything into like one figure, right? As you saw yesterday. I mean, you don't see anything anymore. Having sort of dynamic visualizations where you can turn on, turn off different data sets maybe, or turn off or different tools, and to compare the different tools would make it much easier, right? I agree. I think this is a very important question, but I'm not aware of any tools actually which would allow this.
We're designing tools.
Yeah.
Bio.js is sort of trying to do that sort of
What is
work
it?
over here. Bio.js. So it's
Okay.
sort of just bioinformatics processing in JavaScript, and they have like separate tools to do visualization of genome alignments and that sort of thing.
Okay, Bio.js.
Bio.js.
Should we write it down?
How would you evaluate it is the hard part, right?
Yeah.
How would you say something is better than something else? I think that would be the hard part to...
So example, if something is drawn like a tree and the other is drawn like circles and dots, then it's hard to compare.
Yeah, that's who's the winner is going to be much harder.
Yeah, and another way to go with meta-transcriptome would be probably assembly. Yeah, so how good are the assemblers can produce complete transcript for the gene, which will be probably be a important question.
Gene assembly, meta-transcriptome assembly, that's true. Should we take a note on this?
What
Yeah, look, genes and genes and
gene prediction. I know some people are working on the meta-transcriptome gene prediction.
okay. In terms of priority, do you think what would be should we repeat the challenges we had right now? I think there's still a lot. It's a really active field, right? So I think it would make sense. Or would you say you would prioritize something else above? And yeah.
Personally, I think that doing a second iteration of the same thing would be still beneficial because it's still active and methods are still coming out, and there are clearly from each one of the sections conclusions saying these are good, but it's definitely not the best.
So stuff to be
I
done.
think it's
So
a little bit early.
we would repeat it maybe, and then maybe branch to one or at most two more questions to not get too diverse. Right, and you had another question come up.
Yes, from a participant point of view, it would be good to have some ideas of the metrics that would be interesting to optimize. For instance, when we participated for the first challenge, we didn't know what would be the interesting metrics for you. So if you have some incentive to go towards better sensitivity or better accuracy, then maybe the tool would...
Well, we didn't know either. Right? That's why we had this workshop.
The evaluation metric workshop last year.
Maybe for the results of the third edition, we can...
Yes,
Yeah,
yes,
yeah,
absolutely.
of course.
Yes,
Yeah.
I agree. So for the assembly part, we still wanted, or I don't know how far you've progressed on this, but defining what, well, yeah, for the biological research questions, which metrics are the most important. So yes, in the future we could make this more clear for future rounds.
You could also suggest maybe that some tools use, you could define two tasks. That one that focuses on precision, one that focuses on sensitivity. You could suggest that to participants. So they don't just offer one result, but two results.
Sure, yeah, if we say, for instance, sensitivity is really important for pathogen detection, then you can submit a version of your tool for that, and so on, yeah. So yes, I think that would be an outcome of this first round where we can make that more clear. Okay, so I think now we would start with the Skype talks and with our next speaker, which is Aaron Darling. And then we can, after the talks conclude, so after the two speakers we have, then we can have some more discussions on that, and maybe our picture will have been gotten a bit more complete, or added some more thoughts to that.
So feel free to edit the document, right?